So next up, we welcome our first speaker of the day, Dr. Praveen Vemula. Sorry, before I get into that, one quick point for all the speakers. My colleague, Dr. Meera Thomas, is seated there. She'll be timing the talks, so she'll hold up her cards for your information. And also a quick housekeeping point. Before you take a picture of any of the posters, we request you to take consent of the poster presenter. It's for them to give you the consent. Yeah, so I'll go back to the first talk. So Dr. Praveen Vemula, I'll request you to, oh, okay, sorry. Yeah, he is an associate professor at DBT inStem and Ramalingoswamy Fellow. His lab focuses on developing chemical technologies to solve unmet clinical needs. Additionally, his lab practices extensive science entrepreneurship to take lab technologies to the market and clinic. He will be delivering the first talk of the day titled Biomimicry as a New Path for Drug Delivery, Metabolites-inspired Therapeutics for the Treatment of Inflammatory Bowel Disease. Welcome, sir.
Thank you so much. Can you hear me? Yeah, great. Thank you, Devanshi, for kind of introduction, and good morning, everyone. Today, I'm going to discuss about one of the emerging concepts in the drug discovery, that is biomimicry, and how this could be used for developing potential therapeutics for treatment of IBD. So, but before going to start discussion, I would like to have disclose the conflict of interest, and these are some of the startups we formed based on the technologies we developed in the lab. And I own the equity in these companies, and I actively consult for some of these companies. So my lab at inStem, we primarily focused on clinical translational research, where we run two major programs. One is biomaterials and chemical biology programs. In the biomaterials program, we develop various bioengineering concepts and novel biomaterials to improve the efficacy of the existing drugs. Whereas in the chemical biology program, it's the classical drug discovery program. And the biomaterials, again, they can be two types. You can use them as a prophylactic biomaterial to prevent the diseases to occur, or you can use them as a therapeutic biomaterial which can treat the diseases. In the drug discovery program, again, we have both types of programs where you do the classical medicinal chemistry based new chemical entities we develop, and also novel biologics based drugs also we are developing. But the overall goal of our lab is a need-inspired innovation, where we try to identify unmet clinical need and custom design technologies to solve this unmet need. So although today I'm going to talk a little more detail about our efforts in the drug discovery, I'll take a few minutes and summarize our efforts in our biomaterials programs, what we have been developing for past 6-7 years. So we developed one of these platform technology, disease responsive and disease targeting biomaterials. How you do this is first you identify a biomarker which is overexpressed during a disease, right? This could be an enzyme, cytokine, peptides, and so on. Now you develop drug delivery systems which can be injectable and keep them as a depots in the body. But in the absence of biomarker, they don't release the drug. But when it comes in contact with the biomarker, that acts as a stimuli and release the drug. And the released drug goes and alleviate the disease, that stop production of the biomarker, that stop the release of the drug. In this way, disease can control when to release the drug and how much should be released. So I'll show you how these concepts could be used for multiple medical applications. For example, when you talk about organ transplantation, you can have be it is internal organ or external organs hands and face and limbs transplantation. The post-transplantation, people have to take the immunosuppressant therapies for life, right? But that leads to your global immunosuppression. They become immunocompromised and life that happens. So, but this immune responsive biomaterial can keep as a drug depot. It releases the drug only when the immune activation happens. So because of that, we can protect the transplanted organs through life without causing any global immunosuppression. And similarly, this inflammation responsive material could be injected into the joints, and where we can have a much better therapeutic outcome by reducing the frequency of these injections. And furthermore, we have also shown that such inflammatory responsive material could be used for improving the therapeutic efficacy for treatment of IBDs and so on. So to take this technology further, we found a startup company in Boston which is taking to clinic right now. And another platform technology, we developed this anti-pesticide technology to protect farmers, because these farmers, they spray pesticides without having any protection. It falls on their skin and they inhale. And these are highly neurotoxins. Because of that, they ended up having severe neurotoxicity and death when you have more exposure. So for that, we developed two types of technologies. One is like having this dermal cream. You can apply as your daily cream, but when any pesticide comes in contact, this chemically deactivates them and prevent them the exposure. And the second one is like we also developed this active fabric, which they can wear as a regular dress. But again, this fabric can deactivate all the pesticide that comes in contact and prevent them exposure. And to take this further, like we formed a startup in Bangalore, but right now we are working with the CDSCO to take as a run a clinical trial. So very recently when pandemic happened, like we developed some of the very active small molecules, which are these antiviral molecules, which are covalently attached to the fabric. And this germicidal fabric could be, can deactivate all the virus, bacteria, and fungus upon contact. And based on this, we developed a array of antiviral mask and apparel and so on. We commercialized already in 2020. So these are some of our efforts in the biomaterials program. And that comes to the chemical biology program, where if you think about the traditional drug discovery, there are two major practices what we have. One is natural product based drugs, where you identify the potential drugs from the medicinal plant source and develop them as a drug. And second one is a classical medicinal chemistry approach, right, where either you identify a new target or develop a new target and screen millions of molecules and find the hit and develop further lead molecules. And that's a classical pharma approach what we do, which is also a very investment intense. But very recently there is another concept is being emerged, that is like a biomimicry, right? Because in our body, depend upon our diet and so on, we develop this variety of small molecules which have the variety of biological function. They modulate our biological functions. Although they can modulate our biological function, but they might not act like a typical drug-like properties they don't have. So idea is if you can understand how they are inserting or exhibiting their biological function, by getting inspiration from them, one can develop a potential synthetic molecules. So that's the biomimicry approach. I'll show you how this could be used for treatment of IBDs. So when you talk about IBDs, comes in a Crohn's and colitis, two variants, right? And today if you take, the two classes of drugs are being prescribed, either anti-inflammatory agents or immunosuppressants. But both categories, these are disease managing drugs. They don't modify the disease. Otherwise, in other words, there is no cure for it, right? So that's why there is a huge need to develop drugs which can potentially cure these diseases. And that's why we focused on understanding the microbiome, because as you know, the microbiome, it's a double-edged sword. When you have this, it can have much better therapeutic or beneficial effects, but the dysbiosis of microbiota can lead to pathological conditions as well. And very recently, there's a lot of evidence is emerging where the microbiome can control the function of pivotal organs, right? And it's incredible. The bug that sits in your gut can control remote organ, right? How does it do? So typically depend upon the diet what we take, these microbes convert them into variety of molecules. These are metabolites, and which can exert their function. So if you identify such active molecules and understand what is the mechanism, how they are exhibiting their biological function, one can get inspired and develop the drug. So that's our overall approach. And it is known that when you take phytochemical rich diet pomegranate, cherries and berries, they known to have the beneficial effect. And because of that, when you take this diet, your gut bacteria develop a series of metabolites called erythrins, and people have shown that one of such metabolite called erythrin A has the beneficial effect. And people have established various anti-inflammatory properties of that through in vitro assays. But unfortunately, these results didn't convert into in vivo efficacy. The reason is if you look at this molecule, it's a biphenol compound which is connected through cyclic ester, lactone ring, that keeps your structure planar. But when you oral gavage them, in extreme pH in your stomach, and digestive enzymes can break it, and once this lactone breaks, your planarity lost. So that loses its activity. So we thought can we solve this problem? We started with that. One way to do is develop nanoparticle which can encapsulate and protect from this degradation in pH and digestive enzymes. And in addition to that, if you can targeted manner deliver these drugs to site of inflammation, we might have much better therapeutic efficacy. But if you think about IBDs, like you don't have entire intestine inflamed, right? You have patches of inflammation. If you have a system which can go and specifically adhere to the only ulcerated tissue and release the drug, you might have much better therapeutic outcome. The question is how one can induce such specific adherence to the only ulcerated tissue, right? If you look at the site of inflammation, as a protective mechanism, our body produces a lot of antibacterial peptides and bactericidal proteins to curb the infection. But all of them, if you see, they are cationic proteins or cationic peptides. So if you see inflammatory tissue, it's a cationic charged bed compared to your healthy mucus. We thought, can we use a simple ionic interactions to induce the specific adherence? So if you have a particle which have the complementary charge, which can interact with these cationic peptides and enzymes and bind them and release the drug. So that's the overall concept what we are developing. For that, like we use a series of excipients, and we develop a solid core particles where we can encapsulate its erythropoietin efficiently. And also we have the natural polymer which can coat it on the surface to render the negative surface charge. These are typically like up to 400 nanometer particles, and also have the negative surface charge. Once we have that, we also wanted to see whether are they stable in the GI tract. And if you see, like when you expose them to simulated gastric fluids, intestinal fluids, you don't see any burst release. That means they are stable and they can able to protect from the degradation. And after that, we wanted to see whether they have the ability to bind to the protein that overexpressed at the site of inflammation. So we took transferrin, which expresses the inflammatory site, and coated on the surfaces. As a control, you take the mucin coated surfaces, and dilabeled particles can be incubated and watch them to see how much they adhere. As you can see, like all the particles like adheres to transferrin coated plates, but it's nothing surprising, right? You have cationic protein and you have negative charge particles, so they have to bind it. The question is, the similar level of adhesion can we achieve in vivo conditions? So to test that, like you take the normal mice, and if you give them dexam sodium sulfate for 7 days, they develop the colitis, they have the inflamed colon. Now we can oral gauge these particles, and after 24 hours, you can surgically remove the colon and see how much amount of particles that adhered. As you can see, all the particles, they adhere to the ulcerated colon compared to your healthy ones. Right? So, but if you see the overall whole animal imaging also, when you have particles only retain even after 24 hours when you have the colitis, but in a healthy animals they completely excreted, which clearly shows that indeed these particles have the ability to bind to the inflamed tissue. But these are rodent tissue, right? So that can we get the similar adherence in human tissue as well? So to test that, we collected tissue biopsies from patients, colitis patients. From same patient, we collected tissue from ulcerated site and also in a healthy tissue from the same patient, and ex vivo incubated and tested their ability to bind them. If you see, like all the particles were extensively adhered to the only ulcerated tissue that was taken from the patients, but from the same patient in healthy tissue, they don't adhere. So which is clearly suggesting that indeed these particles have the ability to bind to inflamed site. So once we have that, we wanted to see how this can be related to the therapeutic beneficial. So again, like we developed the colitis in animals, and the drug was given either directly or encapsulating into this particle. And if you see the overall therapeutic outcome, one can measure by measuring the colon length, because when you have the disease, they lose the colon. But when you have this nanoparticle which is completely protected, which is much more apparent in the histology, where you can see in a healthy animals, you have the nice cryptine villi structures. Upon disease, they lose them. But when you have these particles, they completely protected the loss of this architecture. And then we wanted to see how robust these particles are. So for that, like what we have done is, after completely developing the disease, we gave one single dose of this nanoparticle and tested their ability to cure the disease. And again, we can see even a single dose of administration completely protected these animals. And it's almost like a pure phenotype what we are seeing, right? One side, like we were super excited because like the robustness what we saw, but it also equally made us very skeptical, because if you see, these are chronic inflammatory disorders, and we are talking about an anti-inflammatory molecule, right? So how could have the completely reversal phenotype? Because if you see the healthy gut, you have this one single layer of epithelial barrier, and which is tightly knitted with the tight junctional proteins, and that's how it prevents any possible infections. But when you have this disease, chronic inflammatory conditions, you lose these tight junctional proteins, that causes your leaky barrier. Because of that, the commensal bacteria keep getting exposing to the body, the immune system gets activated. That's how these people have persistent inflammation for a long time. So if it is anti-inflammatory, this cannot be reversed. It has to act on the cause rather than this effect, right? So our hypothesis, probably this might be stimulating to overexpress tight junction protein. It restoring the loss of barrier function. That could be the reason why it is doing. So to test that, and also like we also wanted to solve chemical problem, because as I told you, have the lactone ring, right? We just reduced and we made it into cyclic ester, cyclic ether, which is completely stable under pH conditions and extreme pH and the digestive enzyme. So now you don't need any particles to deliver them. You can directly give the molecule itself. So we went and tested, like, upon treatment with these compounds in all epithelial cells, we can see they overexpress series of tight junction proteins such as claudin-4, ocludin, and zonocludin. And through immunofluorescence also we have shown. Then we wanted to see whether we can restore barrier function and reduce the permeability. So that we can do by transfer plates, because you can have the monolayer of cells and you can add your polymer, fluorescence polymer, and see how much it penetrated. And when you induce the inflammation, you can see barrier dysfunction and you see the increased permeability. But upon the treatment with these compounds, it's completely restored the barrier and you see the loss of permeability. Then we wanted to see whether we can achieve similarly in vivo conditions as well. For that, again, you develop the DSS model and treat these mice with the compounds. And one way to measure in vivo barrier function is you oral administer polymer and you collect the blood and measure how much polymer it enters into the body. In a healthy condition, you have the very tight barrier function, right? So you don't see much compound coming into the circulation. But in the disease conditions, you see huge barrier leak. A lot of polymer comes into circulation, but when you have this compound, it completely again restores the barrier function. You see the anything entering into the body. Similarly, again, like it has shown anti-inflammatory properties and also nicely protected the colon function. And not only acute conditions, then we wanted to see in chronic conditions as well, where repeatedly you expose to DSS and have the chronic condition. Even in chronic conditions, you see the barrier dysfunction is pronounced. But when you treated with these compounds, even in chronic conditions, completely restore the barrier function. And also we have shown that in the tissue level also they overexpress tight junction proteins and is anti-inflammatory and completely protected the graft and so on. So then what we have done is like based on this entire data, it's clearly suggesting that indeed this molecule not just anti-inflammatory molecule, but they are stimulating the gut cells to overexpress tight junction proteins to restore the barrier function. So then we wanted to understand the mechanism of these compounds. So we have done a series of RNA-seq experiments and all. I'm not going into detail, but like we proposed the mechanism where these compounds goes through AHR and NRF2 pathway, and that's how they overexpress tight junction proteins. Then we wanted to see whether validate this approach. So then what we have done is like AHR and NRF2, these are transcription factors, right? Upon activation, they translocate into the nucleus. So we tested them. When you treat with these compounds, you can see entire translocation of the AHR into the nucleus. And also when you knock down AHR using siRNA, they fail to produce any tight junction proteins, which is clearly suggesting that indeed AHR playing a role. And similar thing we observed in NRF2 knockouts as well. So these both are important to achieve this efficacy. Then we wanted to see whether that can translate into in vivo efficacy as well. So we have used all the transgenic mice, where if you see the AHR knockout mice, in the wild type, like it completely protects the barrier dysfunction, but when you have the AHR knockout, they fail to repair the barrier, and it failed to curb the colitis as well. Similar manner when you do with the NRF2 knockout mice also, we have the similar approach or outcome. So which is clearly suggesting that indeed these compounds are going through AHR and NRF2 pathway. And this also validated through various other inflammatory disease models as well. And just to summarize, like we started with solving a metabolite problem by using the biomaterial approach, but that led us to completely discovering a new role of these molecules, how they are working. Based on this mechanism, then we wanted to develop further this drug. Also, these molecules have shown fantastic efficacy, but they are not really like a drug-like molecule because it doesn't qualify as all the drug property that's supposed to have. So to take that further, we formed a startup company in Boston and we raised the seed money. Then we have done almost 3 rounds of medcam by keeping the efficacy same and the mechanism same, but inducing or introducing all the drug-like properties. Now we have a lead molecules. We already found the lead molecules, and we have done entire preclinical toxicity and all that's completely completed. Now we are just going to initiate a pre-IND studies, and we are engaging with FDA now. So with that, I would like to stop here, and I would like to thank the wonderful group of people like we did this entire yes. Yeah, the question is why company in Boston, why not in India? We do have companies, 4 companies in India, we put it, but this particular company in Boston, the reason is twofold. One is the funding. In my opinion, I think for pharma-based companies, having that raising early funding is still tough, at least through my network, I couldn't find. Whereas like in Boston, like I think pharma-based startup, we can raise the money relatively easier. And the second one, I also want to tap into the network, which want to have the people like took the molecules right from the concept to the clinic, been there, done that kind of network, where I could find a lot more vast number of members from these big pharma companies and all. So now we started looking for building that network here from probably our next company would be here. Yeah. Regulatory process, I think that comes in further down the road. We are not there yet. Because you see, it doesn't matter whether I found a company here or elsewhere. If I had to go to FDA, I had to deal with FDA. So the regulatory process depend upon where company from would not have much influence. But like till to reach that stage, I think the location matters.
Hello. Very interesting talk, sir.
Thank you.
One question is how long does this show efficacy? How long does this last to show efficacy once administered? How long do you
See, it's depend if it is acute model, and once we give like 7 days, it can completely recovers. After that, you don't see the relapse. Right? In the chronic model, you have to keep giving but when you stop exposing to DSS, that is insult, then it's okay. Then we can take off from the treatment. This can completely recovers from there. So it's depend upon the severity as well, whether you have mild to moderate to severe. Based on that, your therapeutic regime changes.
It is on.
Okay. Yeah, nice talk, Praveen. My question is that your molecule shows very promise for improving the barrier function. And you said anywhere there is inflammation, it can bind to that. So do you think it can work for any other conditions which have similar, or is it only specifically for the IBDs?
No, it's a great question. Like, no, any inflammatory like condition it does. In fact, although we showed IBD, our first clinical application is not IBD for this one. In fact, like we are starting with the bladder cystitis, where you have the frequent urination, but when you have the inflammation, like it's highly painful, and you don't see any retention of the drugs that you give. Maximum they stay like 15 minutes. So, but with our system, like we could improve that retention time more than 24 hours despite of frequent urination and all. So that is our first application what we are doing.
Quick comment and a question. My name is Amjad. I'm from Biocon. So a comment on forming the company in Boston. So one of the reason also what I also noticed that companies which are formed in Boston versus Bangalore or Hyderabad, they have high evaluation at the early stage. So that is also perhaps one of the reason, and it goes favor in the founders. And my question is that are there any AI/ML based models exist which can predict that, okay, these are the normal sort of, gut microbiome and these are the disease gut microbiome, this molecule versus that molecule, and what drug will work better in X versus Y versus Z sort of microbiome? Thanks.
Great question. I think I'm not qualified to answer. I think that Sridhar should be able to answer later about the AI because I'm very ignorant in AI. But there's a lot of, it's in early stage if I believe, like, how people are try to understand. First you have to map extensively, then feed that information into AI system to learn and come up with that. I think it is in early stage, but definitely it would have the, an effect. But I can't tell, like I don't have the
Next up, what are the side effects of these immunities?
sorry.
What are the side effects of these immunities?
Well, for example, our first molecule what we developed, right, which was fantastic efficacy, but like we didn't pursue it because if you see, it has like a 2-hydroxy compounds. It forms like a very nice, beautiful phenol-like structure. So it's immediately it's a genotoxic. It becomes. So immediately it fails. Same test first day itself it fails. So that's where like we took like a 3 rounds of medchem to systematically curb that problem. So right now we have the molecule like which is like very safe. For example, just to give an example, like if you do everyday repeated dose, 2 kilogram per kg dose for 28 days, you don't see any toxicity at all. So that is the level of safe profile what we have.
Hi, nice talk. But I just wanted to ask whether did you investigate it into any other oncogenic conditions, for example, colon cancer, which is also associated with inflammatory mucus?
Yes,
In the gut.
yes, we did. We just one paper came out in Theranostic. We just published that. So, in fact, especially colorectal cancer, your inflammation is the starting point, and it breaks the barrier. And under normal condition, like any genotoxic material, if you get exposed, if you don't have barrier dysfunction, you can still escape. But when you have the barrier dysfunction, even minute amount of genotoxic material can induce that. So what we have shown is like if you combine with the chemotherapy with these compounds, we can completely cure them, or like which have much better therapeutic efficacy in colorectal cancer treatment.
Nice talk.
Thank you.
So, two related questions. One, does this molecule work or is it inactivated? How this molecule is show that inflammation so that come up to affect it. The protein I understand can induce the expression of this. This is the generation of it.
So to answer your first question, I'm not sure it is altering anything microbiome at all. It doesn't act on the microbiome. But again, like saying that, like we haven't looked at it, but we don't see any reason to how this molecule can alter your microbiome itself. Because microbiome, I think, probably various other factors can influence. Regarding the second question, and in fact, we have a lot of evidence. We just identify like one is expressed adjunctional proteins, but it also increases the mucus secretion. And if you see the mucin production is far higher when you treat with this compound. So because your mucus is act as a glue in the beginning, right? So then it acts as a 2-level. One, it increases the tight junctional protein so that you stitches back, and it also increases the mucin production, which have the gum layer on the top of it. I think cumulatively that it has a much better regeneration capacity. But since you brought this one, I wanted to add this, like, now that we understand this mechanism, now we are developing another next stage of material molecules, which not only induces the mucus production, but also stimulate your gut residing stem cells. so that you can increase the proliferation of your stem cells to restore and regenerate entire gut. So that molecule already is there, but like a bit early stage, I would say.
But Praveen will be around.
Yeah, sure.
A fantastic talk, I mean, very inspiring.
Thank
So what I saw is that everything leads to a very nice high quality publication, but then it also gets translated through a company. And I was wondering what kind of support or ecosystem in inStem enables this. Can you tell us a little bit about that?
Yeah. Oh yeah. So because inStem contribution, I can go on. So to answer that, yes, we have a fabulous ecosystem, developed, established at inStem and the Bangalore Life Science Cluster in general, because we have next door we have NCBS and the CCAP and so on. So of course, science like it produces from everywhere. So one of the thing in early stage what we realized is if you want to translate them, one has to practice robust entrepreneurship programs, and having that policy framework is very important. So in the very early first couple of years itself, inStem has set up this entire entrepreneurship policy framework. Now it is robust enough that if any researcher, PI or student, anyone wants to go and start a company, within 2 weeks we can do entire paperwork from them to get the permission. So that's the level of support what we get. And again, having CCAM next to it, it gives a lot more access, very informal conversations and all, people can get exposed, because one of the things in science entrepreneurship what we see is just getting that confidence that okay, this can be done, and those conversations can happen informal in these places, that will allow us to trigger them. So of course ecosystem has a great role in it.
Thank you, Dr. Praveen Kumar, for that fantastic talk. Request everyone to give a round of applause for the great start to the event. I'll request Professor Abhijit to present a memento to the speaker. We're now off on to our next talk. Welcome Dr. Preeti Varke on the stage. She is the Director of Cell Biology Division and heads